So welcome to the new session. So far, what I have tried to do is to discuss with you general aspect of biotechnology, looking at the historical perspective, what is done in biotechnology, and also I gave you an overall view about the organization of the unit of life, that is the cell, how it organizes itself, how it looks like externally and internally, the kind of organelles it has, and what are the functions of those organelles, and what is the composition of the cell, chemically, what it is made up of, and we talked about the organization of the genome, how it organizes itself, and also we looked at that how the genetic complexities arise in the living forms and from prokaryotes to eukaryotes, and then the genetic basis of variation, linking trait, phenotype to genes, to proteins via RNA, the current understanding about the Mendel's concept in today's era of molecular biology and biochemistry, and how these genes from prokaryote to eukaryote express themselves in a regulated manner, you know. So now I feel that you have the basic information about the cell, which is the basis of life and all living systems. Now what I'm going to do is to take you beyond this and very specifically talk about the plant biotechnology. Okay, so biotechnology, as we discussed, is basically the application of biology or biological principles to modify organism or part of organism for driving or products or services for the betterment of mankind. But when it comes to plant biotechnology, actually, it very specifically involves or revolves around two things. One is plant tissue culture. So again, a technique which has been used for cloning purposes. Cloning in the sense, very specifically, to generate clones of the same plants, you know. So not in terms of recombinant DNA cloning. So plant tissue culture techniques and the DNA introduction, or what we call gene transfer, you know. So, and both these aspects of plant biotechnology are interconnected, actually. Okay, so we will be one by one talking about both these aspects of plant biotechnology. First, I'll begin with plant tissue culture and how this culture has, or plant tissue culture has been utilized for improving the plants for their performances, whether it is towards productivity, quantitative or qualitative. And then the DNA introduction or the gene transfer basically means genetically modifying plants for a particular reason, whether we want plant to perform better in the field against the abiotic or biotic factors, and also qualitatively to improve the product of the plant against, for example, this gene transfer involves the gene identification for a particular process. It can be stress resistance, herbicide resistance, viral resistance, bacterial resistance, fungal resistance, insect resistance, or improving the nutritional value of the produce. Okay, so this part we will see subsequently, but we'll focus on the plant tissue culture aspects. So, what is plant tissue culture? Basically, if we go by definition, or if we have to define, okay, so basically it is the culture of seeds, tissues, cells, protoplasts on a nutrient media under sterile condition. So basically, we are trying to culture or grow the organs, tissues, cells, protoplasts on a nutrient media under sterile condition is called plant tissue culture. Okay. Now, this actually involves three fundamental abilities of plant cell. One is the totipotence. Now, it has been observed or found out that plant cells are totipotent. What is this totipotency or totipotent nature of plant cell? Means that each and every somatic cell of a plant is capable of regenerating into a new plant. See, normally the plant reproduction or in eukaryotes, the reproduction takes place through sexual means, through sexual reproduction. But here, the totipotency or totipotent nature of plant cell is that each and every somatic cell, not the reproductive cell, somatic cell is capable of giving rise to a new plant, provided the right kind of growth conditions are provided to the cell. Now, this is very important clause, that a cell should be provided with right kind of the nutritions as well as the growth factors which are required for a cell to get differentiated into the whole plant. So, one factor, fundamental ability of a plant cell is the totipotence. That is, the somatic cell is capable of regenerating into whole plant. And then dedifferentiation. What is this dedifferentiation? Plant cells, the cells, they undergo differentiation to form stem cells, leaf cells, or shoots, or flowers. So this kind of differentiation takes place among the cells while developing. But when we take any tissue from a differentiated part of the cell of the plant, like shoot, root, flower, stem, it undergoes dedifferentiation. Means these cells, they undergo dedifferentiation to form a mass of cell, what we call callus. So this is another ability of a plant cell, that it can undergo dedifferentiation. Again, the growth conditions have to be appropriate for cells to survive and undergo dedifferentiation. And then this differentiated cell or mass of cells, what we call callus, is capable of redifferentiation into the organs, again, like shoots and roots, and then later on flowers and seed set. Okay, so these ability of plant cell to undergo dedifferentiation and redifferentiation is also very, very interesting aspects And then third is the competency. Now, competency is the endogenous potential of a cell or a tissue to develop in a particular way. I mean, the tissue cells are competent, or they always have an inherent endogenous potential, to grow or develop into a particular way. So these are the three fundamental abilities of plant cells which gave rise to this area of plant tissue culture. A, to dependency, and two, de-differentiation and redifferentiation, and then the competency of the cell, or the cell has to be competent to undergo a developmental process in a particular way. Okay, so if we see the historical part, of plant tissue culture, it all started with the cell theory, which was given by Sheldon, that cell is autonomous and totipotent. And then first attempt for plant tissue culture was made in 1900 to last century, and then in around 1939 or 40, the continuous growing callus cultures were produced, and then the whole plant was developed in the middle of the last century using shoot tip as the explant, and in around same time the organs were regenerated on callus, so a differentiated de-differentiated mass of cells like callus could be regenerated into organs, and then it plants from a single cell, not from the mass of cells, but the single cell could be regenerated into whole plant. And then in 60s, not cell, but as you know, the plant cell has the outermost covering as the cell wall. So in 1960s around, this cell wall was removed, and a naked cell was generated by removing the cell wall. We call that as protoplast. So protoplast is nothing but plant cell devoid of cell wall. Okay, so the methods could be regenerated to produce protoplast or a plant cell without cell wall, and then around the same time, the more defined synthetic media was developed by Murashi and school, which had all the components required for plant cell to grow in vitro. So a very well-defined media. And after that, there's millions and millions of compositions now available. And then the first application of plant tissue culture came in the 1964, where clonal propagation was achieved. Means taking this the somatic tissues, the plants could be regenerated, having the same genetic makeup, what we call clonal propagation. And again, around the same time, it could be possible to generate haploid plant, using from pollens or the haploid cells, first from pollen and then from ovules also. So, and that revolutionized the agriculture side, actually. So haploid productions, and then in the 70s, was the era where the protoplast could be fused, and the hybrid plants could be generated with a view to generate plants which will have which are generated by fusing two genetically different cells, or from two different plants, which otherwise is not possible through sexual reproduction. Sexual reproduction is limited to species, but here the two somatic cells from two different species or genus was taken, like for example, one from potato and another from tomato, and with a view to make a plant which will give potato under the soil and the tomato in the top, and success were achieved to generate a plant which was named as tomato, and we'll talk about this later. So these kind of work was done in the 70s of last century, and then plants also could be regenerated from this protoplast. So plants could be regenerated from single cell, the whole plant, and from protoplast to cell to whole plant. Now, this was very interesting discovery, and it opened up an era of possibilities where if you modify genetically single cell, in this case protoplast, then one can generate genetically modified plant. So it's a question of just modifying one cell or protoplast and then regenerating whole plant, which is genetically modified. And before this could be achieved, this discovery of protoplast regeneration was exploited for picking up somoclonal variants to to generate plants which are better performers than their parental plants. And I'll talk about that later. So this is, in brief, a historical account of how the plant tissue culture area or field has grown over a period of time. Now, what is the basis of plant tissue culture that we have to understand? Okay. So basically, there are two hormones which affect the plant development. One is auxin and another is cytokine. Okay. So auxins, they stimulate root development, and cytokine stimulates shoot development. And what is seen that it is the ratio between auxin and cytokine which decides the path of cell differentiation. For example, what I'm trying to say is that if the auxin concentration is more than the cytokinin, then cell tend to differentiate into root system. Cell tend to produce roots. And if it is other way around, that is cytokinin is more than auxin, then the pathway is diverted towards shoot formation. So basically, in vitro, it is this ratio between cytokinin to auxin or auxin to cytokinin which decides the path of differentiation, that whether the cells are going to produce shoots or roots. And if the auxin and cytokinin are present in the same concentration or equal concentration, then cell tend to undergo division only. So it only multiplies and produces a mass of cell, what we call S. Okay, so this is the basis of plant tissue culture. When this understanding of role of hormones was realized or studied, that these two hormones are responsible for cellular differentiation, even normal plant growth and development cycle. So when the seed grows and cells, those who are committed for root development has cytokinin to auxin ratio in a particular way, and those cells who are committed for shoot development has different. And this was very important discovery as far as plant growth and development is concerned. So more pictorially, what is seen here, that how they control auxin and cytokinin. So if they both are same, then you have, if the auxins are increasing, cytokinins are less, then you have the shoot formation. And if shoot formation, if the cytokinin is more, auxin is less, you have shoot formation, and other way around, if you have more oxygen, less cytokinin, you have root formation. So normally what happens, you first have the shoot formation and then change the media to more of oxygens to give you the root system. So that's how in vitro the culture is controlled, you know. But if it is the same, then you will have the callus formation. If the same concentration of oxygen to cytokinin is same, then you have the callus formation. So this is what happened. So when the in vitro, the cells are undergoing de-differentiation, you see the callus formation, you know, and then the shoot formation, and then when they change media, you have the root formation, and then this plant is transferred to the pots or the soil. So that's how in vitro the whole process of culture goes on, and it follows two paths. One is via organogenesis or through somatic embryo formation. Organogenesis means first the callus is formed, and then the shoot is formed, and then root is formed. But through somatic embryo formation, the callus produces pseudo-embryo-like structures. They are not the actual embryonic structure which are produced after fertilization, but this somatic embryo, which is produced, is from the somatic cells, callus cells, which is capable of differentiating into shoot system and the root system. So in vitro plant culture follows two paths of differentiation. One is through organogenesis, that is callus, and callus is differentiated into shoot and then to root. And another, the callus forms embryo-like structure, what we call somatic embryos, and these embryos then germinate or grows into shoot and root system. The whole plant is generated. So these are the two ways by which this is. Now, what is the technique actually? I mean, you can have the plant, you take the explant or the single cells, put it on a nutrient media with growth regulators, and you have the regeneration of the plants, and then the plants are taken to pots. But then, how it is achieved as a technique? What is done? That is very interesting to know. And very briefly, I will go through this technique and the salient feature of this technique, that how it is achieved. So in this, if you see, there are four different factors. One is the growth media, another is environmental factors, and then the explant. Explant is the source, I mean the tissue with which one starts, and then the genetics of the mother plant or the source plant. So these are the four factors which play a very, very crucial role in plant culture technique. So if we talk about growth media, basically, so even this has undergone its own evolution. So starting from ill-defined media, like using the plant extracts, you know, as the new source of nutrition, to well-defined media in 60s, as I told you, Murashi and his group first introduced the well-defined media. And what I mean by well-defined media, it had all the components which a living cell requires as a component. So it has minerals, growth factors, carbon source, hormones. You know, and minerals means the macronutrients, micronutrients, you know, and growth factors means the hormones, auxin, cytokinin, gibberellins, FCK acid, the other, I mean, but the major were auxins and cytokines, and the gibberellic acid. Carbon source, which is very, very important for a cell which is not photosynthesizing, so it requires source of energy, sucrose, And subsequently, other sources of carbon have also have been used, and then the environmental factors, like light, temperature, photoperiods, sterility, uh, you know. Uh, so these are the very, very important factors which play a very crucial role. And then the expert. So what is the, usually it is the younger tissues which are more uh, amenable for tissue culture regimes, and they are considered to be better for tissue culture. But theoretically, if you see, as I mentioned, that each and every somatic cell is capable of regenerating into whole plant, but that doesn't happen practically. So these are the factors which play an important role, that what is the younger source or younger explants better show better regenerations as compared to the mature and the older plants. You know. So, and also what is the genetics behind, means in a genus, the different species, they are more, first of all, the different, among different genus or different types of plants, some plants are more amenable for in vitro cultures or manipulations as compared to others. Within the same genus, some species are more amenable as compared to other species. For example, if you take rice, so we have rice, Japonica type rice, Indica type rice. Japonica type rice is more amenable for tissue culture manipulations or in vitro manipulations as compared to Indica type of rice. It is tough to manipulate indica type of rice in vitro. So this is just to give an example that how genetics also plays a role towards the success of in vitro manipulations of the plants. Yeah. Now, this well-defined media and Marashi and school perceived, now the basis was the information, knowledge generated on the plant nutrition, what a plant requires, you know. So taking clues from that side, uh, the Russian school media had the major elements and the minor elements. And how this major and minor were classified, that is based on how much the cells require. If an element is required in higher concentrations, higher amounts, it was grouped into major components or major elements. And if an element is required in minor quantities or lesser quantities, it was put as minor element. Okay, so clear-cut division of major and minor. So these are some of the examples of major components of the synthetic media. And also, which you see in these tables, that if a plant is deficient for that element, it shows the deficiency symptoms. So it means, and which goes when you give externally that element to the plant. So for example, what I'm trying to say is iron. Here, if you see iron deficiency in plants shows you chlorosis or yellowing of the leaves. So if you reduce iron from the media, you know, keeping all other elements same, then the plants show you chlorosis. So that's how it was concluded that iron is major component of plant nutrition. So like that, calcium, magnesium, nitrogen, phosphorus, potassium, sulfur. These are some of the major elements, which are required by the cells, and they have there in the media as a major component. And then you have minor elements like boron, chlorine, copper, iodine, manganese, molybdenum, zinc, you know. And again, the deficiency of this gives you the deficiency symptoms, and the plant is not healthy, you know. That goes for major and minor both, you know, and therefore any media which is being used for growing a plant tissue cell, you know, should have all these constituents of the media, you know. And then, so in addition to these major and minor elements which are required by a plant for its growth, you know, are taken in a media to grow the cells or the tissues of a plant in the in vitro. Okay. In addition to this, they should have a source of carbon because the cells in culture, they don't photosynthesize, they can't make carbohydrates, so they have to have a source of carbon. And predominantly, and what is used is sucrose, you know, as the carbon source. But as a variant, there are other carbon sources also have been tried, which you will see in different papers, mannitol, sorbitol, and hexitols. These are other complex carbon sources which have been tried and have been successful or successfully used for. Then vitamins is a very, very important component of the media because like animal system, plants also require vitamins for their metabolic processes. So a variety of vitamins have been tested, used, you know, and then you have growth regulators, which have to be there. So you have auxins, so you have natural occurring auxins and the synthetic auxins like 2,4-D, you know, which are 2,4-D and all as to the cytokinins, you know, cytokinins auxins, which have been, and these are different lists is there, you know, which have been used for driving, you know, for directing the differentiation towards either callus formation or the root formation or the shoot formations. And then you have gibberellins, abscissate, and in some places even these hormones have been used along with cytokine, kinatin, or to direct the differentiation process. And then you have other gelatin agents like I mean, you have to have the media is not liquid, so it has to have the gel formation to give a sort of a soil-like of an environment to the growing and differentiating tissues. So that is provided by agar-agar. You know, so that is used here like this. So it gives the support to the growing plant. And then in some media, amino acids have been used. Although antibiotics are forbidden and they're not being used, but in very rare circumstances where the explant is coming from the highly infected source, one can use these kind of fungicides or bactericides. But that is very specific cases these antibiotics are used. You know. And then some of the media, they have the natural components which induces the growth and development process like coconut milk, yeast extract, malt extracts, you know, tomato juices, but they are avoided actually. They are not used, but sometimes when these things have helped in getting in vitro differentiation. But it is always preferred that one uses the known media where we know what that what we are adding and what it is, what kind of response it is giving. That it is for the purposes of reproduction, that we should be able to reproduce what one is getting. The other person also, if he uses the same thing, should get the same response in vitro. And, you know, if the use of these kind of materials like coconut milk, potato, etc., you know, it depends upon from where the tomato is taken, what is the nutrition value of this tomato or potato or coconut milk, you know. So that may vary, and that reproducibility of usage in terms of responses may be in question. So they are being avoided. But here, if you are working to have an in vitro response, and if you are getting that response, one can use and then make a product. You know, what one is interested is in making a product. So, but then it is avoided. Very important aspect of this technique is aseptic. Means everything has to be sterilized. Means starting from media to the glasswares, other tools which one is using has to be sterilized. The explant has to be sterilized because you grow, you're dealing with nutrient-rich media. If the infection goes inside, then your explant will not grow, rather than other things will grow. The bacteria will grow, the fungus will grow, you know. So it is very, very important that we disinfect or sterilize everything which is being used in this whole operation, you know. So sterilization is to destroy the living matter, disinfectants and chemical agents, and the sanitation, all these things. I will go into the detail of how the aseptic conditions are maintained while doing in vitro cultures. So, for example, if you have to sterilize the explant, the tissue which you want to use in vitro, then chemical sterilization is done by using sodium hypochlorite or calcium hypochlorite, hydrogen peroxide. You know, these are silver nitrate, mercury chloride. So, but then they have to be, I mean, how long is to be used needs to be optimized because they are very, very lethal chemicals. If you expose your explant longer than the permissible time, then the explant tissue will die. So therefore, optimization is. But what they do good to the explant is that they kill the other microflora coming in the explant. You know, the bacteria, the fungus. You know, they are all killed. So that is how the explant sterilization is achieved. And then you have sterilization. You have physical methods and the chemical methods. You know. You have nowadays the whole bench where the operations are done it has this dry airflow, what we call laminar flow chambers, you know, and then the air comes through filters which removes all the bacterial contaminant from the air, so you can freely work, you know. UV radiations you can leave the whole thing UV under UV for some time before you start using. So that also takes care of killing the microbes, you know. So like that, there are various ways, both physical and chemical, which have been successfully used for getting these sterilization done. So, to summarize, what I have tried to tell you is, and which you should remember at the end, is that the cell theory aspects describing the fundamentals of the cell, you know, and then the callus tissues, that is the dedifferentiation part of it, the totipotency of the cell, plant cell, that each and every cell, somatic cell of a plant is capable of regenerating into a whole plant. And then the media components and the sterilization component. Now, these things you are going to do it in the labs also. When you come for practicals, you will see that how the tissue culture is done in vitro. So, following these techniques, you know, so This all has been achieved. Culture of intact plants, seed and seedling cultures, embryo culture, organ culture, shoot tip, root, leaf cultures, anther culture, callus culture, cell suspension culture, protoplast culture. So all these things have been achieved. When I say that, there are plants where all this is possible. You can regenerate protoplast, you can regenerate anther, you can do shoot culture, root culture, leaf culture. Theoretically, yes, all the plants we should be able to show, we should achieve this. But practically, it is, I mean, if you go by a car, fabrication of a car, people get stuck with something or the other in some plant or the other, or they have to struggle a lot to optimize conditions and to provide conditions which the cell requires or wants for uh, getting differentiated into in vitro. So a lot of optimizations have to be done to achieve these cultures in vitro. And the application part of this plant tissue culture technique is enormous, actually. And we will see one by one, for example, micropropagation, that how this technique has been used for multiplying the plants or to generate same kind of clones genetically, you know. So what we call micropropagation, and then the germplasm preservation, somatic variations, dihaploid production, protoplast fusion, secondary metabolite productions, and finally the genetic engineering. Okay, so these are the applications of or biotechnological application of plant tissue culture technique, where the products were developed from, in order to improve their quality in terms of their production and the, and the quality of the produce which they are making, not only quantity but also quality. Okay, and that is the one objective in biotechnology, to modify the organism or part of the organism to make for the benefit of mankind. So we will, in subsequent lectures, we will take one by one and go through that, how the application, what are the, what actually we mean by these, and how they have been successfully employed to drive products. So thank you very much with this, and we will we will be dealing with these aspects in